Hi everyone, welcome to the second video in DNA replication, transcription, and translation. We've already covered this. It's just kind of a placeholder, a title screen. We're going to move on. This particular video is going to be talking about a function that DNA has. Because of its structure, it's able to make copies of itself. And that happens during the S phase of the cell cycle. So just a quick reminder of the cell cycle. Got some growth going on here, but then everything stops right about here, and the cell spends quite a bit of time in the S phase before it goes into another growth cycle and gets ready for mitosis. Now I'm showing you this because I want to remind you that at the end of the cell cycle, one cell started here and it went through the entire thing, grew and matured and everything, and right here at the end in cytokinesis it splits into two. So the point here is that we now, if we're going to make two cells, we have to have two sets of DNA. And so this process of replication is how the DNA can make a copy of itself. This is one of the functions that Watson and Crick discovered after they discovered the structure of the DNA molecule. They realized very quickly that it was going to be able to make a copy of itself. It's unbelievably brilliant, okay? That this is able to do what it does. It's all based on the idea of the complementary pairs. If you remember, we covered this in the last video, that the nucleotides on one side are complementary paired to the nucleotides on the other side, and because of a guy named Chargaff, we know that there is an equal number of guanine as there is to cytosine and adenine as there is to thymine, and then we discovered that they actually bond together while the DNA molecule is connected, or what we call zipped up. In order to do this process though, it has to be unzipped, and for it to unzip, we actually have to have a zipper, and in this case this is an enzyme. Now remember, whenever you hear the word enzyme, I want you to think of cut and paste, because enzymes either cut bonds or they make bonds, and in this case we have two enzymes and each one of them is doing one of the, either one's cutting, one's pasting. For instance, this is helicase. It kind of attaches way back over here and it starts moving down the DNA double helix, and its job is to break that weak hydrogen bond right there to kind of expose both sides of the double polymer. And then along comes another enzyme called polymerase, and these nucleotides are just floating around. They're just laying around. This polymerase acts, this is the paste part. It'll grab, hopefully, the correct enzyme, or I'm sorry, the correct nucleotide, and stick it back together. So polymerase says, oh, there's a T, I better find an A and stick it there. Now there's no thinking involved here. It's not like there's a brain, but this is all chemical. Remember, there were 3 bonding sites on the A's, on the G's and the C's, and only 2 bonding sites on the A's and the T's. So as the polymerase grabs these nucleotides, and they're literally just sitting right there, it's pretty easy to put the right one there because they bond specifically. So the polymerase moves along here and is making a complementary copy. By the way, it's doing the same thing over here. And at the end of this whole thing gets totally unzipped and a new copy is made. And if you'll notice right here, this is a full double polymer DNA. We're going to make a full double polymer DNA here as well. So here's what it looks like. It starts to unzip. You can imagine there is a DNA helicase enzyme unzipping it, and then along comes polymerase and adds the 2 new sides, and as it keeps going up this way, essentially we're going to end up with two new DNA molecules. Now since this molecule is very long, 3 billion of these little pairs, you can't start at one end and work your way down all the way to the other. You have to do this at several sites. So this is done several times along the DNA strand. It's not just starting at the beginning and work your way through. It would take days, months to do this, and we literally need it done in minutes. So it gets done a lot faster. It's a lot more complicated than I just explained it. And I don't really need to go into all that complication, but as you can see, by the way, you see how I was like a bubble? This will keep moving this way, and then somewhere way down here, back over here, something is doing it, and eventually it'll get to here, and they all just kind of work their way across, and this will stop when it gets here. This one will keep going until it hits the next bubble, etc. Okay, I could teach you all this. Remember, we talked about 3' and 5' in the last video. That's important here because on this side it can copy in a continuous strand, but on this side it has to copy in fragments. They're called Onozaki fragments, but yeah, you know, I just don't feel like that's tons important for you to know. I would like for you to know that they don't copy the same. One side copies continually, the other side copies in fragments, but that's another day. But at the end, we are creating, and here's those bubbles I talked about. At the end, though, we are creating two new copies of the DNA helix. Okay, a bubble opens here and it's going, another bubble opens here, it's going, and they keep. This one keeps going in the right direction. This one they're both moving in the same direction. Eventually, this one will get here and then stop, and then another one's coming in from the other side. And so ultimately, much faster than it would if we just started on one end and worked our way all the way across. These bubbles open up and it makes the process happen much more quickly. Literally 3 billion pairs of DNA are copied in a matter of minutes. Just to give you an idea of how fast that is, if you were to watch these DNA, if you were to count these one per second, it would take you 75 years without stopping to do it. That's how huge it is. If you did one per second. So this has to go a little bit faster than one per second because we can't do it in 75 years. We have to do it in about 75 seconds. So it's incredibly fast. And then on top of that, it has to be very accurate. So the accuracy involved here is a couple of things, but the chemical nature of it keeps it from being inaccurate. Now, in 3 billion 3 billion pairs, if you make a mistake, it's going to happen. So after this strand is totally copied, some things come along and check just to make sure that it was done right, and they fix any errors. So even at that, there still ends up being about 3 to 5 errors every time you make a copy. Fortunately, most of the time, that's in a place where it doesn't have any effect. But errors equal mutations, and we don't want mutations necessarily. Because mutations don't lead to being able to climb up walls or have x-ray vision. Usually mutations lead to death. Usually if it kills the cell, that would be better for us because the cell just dies, because if it doesn't kill the cell, usually that means the cell is going to become cancerous. So we don't want errors to happen. We know they're going to happen, but we want the errors to happen in the blank spaces. There's a lot of blank spaces on the DNA where it's just gibberish. In between the genes, there's blank spaces, so that's usually where it happens. Also, we'll talk about mutations later on. They can happen in a place that is not necessarily going to cause a change. We'll talk about mutations later, but just to be clear, these bubbles start all along the DNA strands. By the way, there's not just one big long strand, there's actually 23. So we also, it's happening on, instead of one long piece of DNA, you've got 23 pieces of DNA, and so that makes it where we can copy it faster as well. A little bit of practice just so you can work with me here. I need you to be able to do this. If there's a G here, you notice how G's bonds with C's and A's bonds with T's. You can see all the bondings here, and you can imagine that this is it opening up, and this G and this C were bonded together, this T and this A were bonded together, but that helicase is opening it up. So you can imagine the polymerase coming along, and what's it going to want to stick to that G? What's going to go right there? A C, and if you'll notice there's a C down here, and if there's a C here, it's going to want to stick a G there. If it's an A, it's going to want to stick a T. If it's a C, a G, a T, okay, etc. By the way, the same thing's happening along here on the bottom, kind of the opposite. If there's a C here, it's going to want to put a G there, and if it keeps doing that, notice that you'll get that new strand. I should have drawn a purple line across here. You'll get that complementary strand. Notice that this side, GCACTG, that's the same as what was already up here, GCACTG. So we do make an exact copy. And this is all happening in the S phase, and then when we get to mitosis, one of these copies is going to go into one of the new cells, and then one of these copies is going to go into another one of the cells. And as a result, we now have a full set of DNA, or all the DNA copies are put into each cell, and that's the function of replication. Let's do a little bit more practice just in your head. I want you to pause this and just in your head kind of work your way through here, and tell me what would go here, because this is a pretty complicated thing, and it has to happen very fast. Imagine how long it would take you to do all these. Probably what, 30 to 90 seconds, and this is maybe, I don't know, 1 2 3 4 5 6 7 21. It's about 85 it's about 85 pairs, and it takes you about 90 seconds to do it. Imagine if you had to do all 3 billion pairs in the same amount of time, how fast you'd have to go. I gave you a little key here to help you remember the base pairing rules. But long story short is, if you had to go through and do it, and I want you to pause it, so hang on, pause it. Pause it and do it in your head, and then when you're through doing it, unpause it. Okay, so pause it, and welcome back. So I'm going to give you the answers right here. A bonds with T bonds with A bonds with T, C bonds with G, A to T to A. Now it's not that hard really. I kind of had to every once in a while cheat a little bit because my spacing wasn't working with the new font. But the A goes with that T and that T goes with that A, and there's no reason they're clumped together except just stupid computer. But happened there too, and it happened there. Coincidentally, I didn't have to do it on the bottom. Don't know why. But as you can see, you can go through here and do that. And honestly, I did this pretty fast, and I don't think I made a mistake, but look and see. And if you see a mistake, tell me. All right, so that's how replication works. The idea is, of course, let me go back up here a little bit. The idea is, of course, that we start with one strand of DNA and we're finished. We have two strands of DNA identical to the same, and each strand goes into one of the cells at the end of the cell cycle, at the end of mitosis during cytokinesis. And it's only because of the structure of the DNA molecule that it can do this. So that's going to end video 2. Pretty short. Might want to watch it twice. Replication isn't that complicated. I mean, it can be. Where's that? Yeah, I mean, we can make it really complicated, but we're not going to. Just understand that it opens up and it makes a copy. And I do want to tell you that we're going to be talking about something similar to this again, except instead of copying the entire thing, we're just going to copy a small thing. So you may see something that looks like this again, but it's not the same thing. It's called transcription and translation, and that's going to be the next couple sets of videos. But this ends video 2. Thank you very much. See you at video 3.